Here we have Oscar Rodriguez, and he's got his slides up. Also from the University of Louisville in the Watson lab. And Pam just mentioned that the AIR community meeting is in the chat, and it's 9 o'clock, so let's get started with Oscar, please. Or top of the hour.
Okay, great. Thanks, Felix. Okay, so today I'll be talking about how the
Can't
genome...
see Oscar. Sorry to interrupt. We can't see you, Oscar.
Oh,
Thanks.
do you see me now?
Yes, thanks.
Okay. Yeah, so today I'll be talking about how the genome sets up the antibody repertoire from early B-cell development. And so within the immunoglobulin heavy chain or IgH locus, there are 47 functional variable genes and 27 functional diversity genes and 6 joining genes. And during B-cell development, a single V, D, and J genes, J gene are selected during VDJ recombination to form the heavy chain of an antibody. And as Melissa said, we recently demonstrated that the differences in the frequencies of the V, D, and J genes that are present between individuals, we found that those frequencies were associated with IgH genetic variation. And so, for example, individuals with the T allele at this SNP had higher levels of the gene IGHV366 in their antibody repertoire. And we observed this genetic effect on the majority of the IgH, V, D, and J genes. However, our results lead to a conundrum because, as we know, there are cellular mechanisms during B-cell development, including central tolerance, that remove or edit B cells, and thereby could alter the antibody repertoire. This implies that the genetic effect that we observed in the periphery could be affected by B-cell development, or alternatively, that the heavy chain antibody repertoire is not being affected by these selection processes. In 2003, Waterman et al. Showed that 55% of the developing B cells were autoreactive, but this dropped to 5% of the 5% by the end of B cell development, indicating significant changes to the antibody repertoire during B cell development. And early transgenic mice studies showed that the antibody repertoire was being altered by receptor editing, where the light chain is altered to remove its autoreactivity. And critically, this would only affect the light chain antibody repertoire and not the heavy chain antibody repertoire. However, in 2016, Martin et al. Sequenced the heavy kappa and lambda antibody repertoire of pre-B, immature, and naive B cells and showed that the heavy chain antibody repertoire changed more than the light chain antibody repertoire during B cell development. This would imply that receptor editing is playing a minimal role in contrast to previous transgenic mice study. However, one limitation was the limited sequencing depth of the antibody repertoire. And furthermore, the IgH genetic variants that we identified to be associated with inter-individual gene usage differences at the periphery were enriched in genetic regulatory regions associated with chromatin structure and VDJ recombination. This is critical because VDJ recombination occurs early in B cell development at the pro-B cell stage. And therefore, in order to observe this pro-B genetic effect in the periphery, there would have to be minimal alterations to the heavy chain antibody repertoire during B cell development. And so, given these conflicting data on how the antibody repertoire changes during B cell development and the significant effect of IgH genetic variation on the antibody repertoire, we sought to understand how the antibody repertoire develops in the context of both B cell development and genetic variation. And so in this study, we sequenced the antibody repertoire from developing B cells along with the immunoglobulin loci. And this work was performed with Dr. Ranjan Sen from the National Institutes of Aging and Dr. Zhang Qiu, who's now an assistant professor at Chongqing Medical University. And so for 6 individuals, we fact sorted pro-B, pre-B, and immature B cells from the bone marrow. And we sequenced their antibody repertoire along with the antibody repertoire for naive B cells. Additionally, we sequenced the heavy kappa and lambda loci for each individual using long-read sequencing. And so from this long-read data, we were able to resolve structural variants and accurately determine which immunoglobulin genes are present within each individual. For example, on one haplotype in individual D, we identified a novel structural variant that contains 40 kb of novel IgH sequence and four novel functional genes. On the alternate haplotype, individual D has 100 kb sequence and 8 genes deleted relative to haplotype 1. And in fact, within the structure variant hotspot, all individuals have a different set of IgH genes. Using the long-read sequencing data, we were also able to resolve all the gene alleles. In this tile plot, the x-axis are the V, D, and J genes for heavy kappa and lambda, and each row is an individual, and the color corresponds to the different alleles. For example, 4 out of the 6 individuals have the allele star 02 for the gene IGHV373, which differs from the IGHV373 allele star 01 at framework 1. And so what's clear is the extensive amount of allelic diversity, the gene allelic diversity. Individual E has 241 V, D, and J alleles from all 3 loci, which is 41 more alleles than individual A. Additionally, the empty spaces in this plot correspond to deleted genes. Here I'm highlighting the structural variant hotspot I previously showed. And so clearly, given that the VDJ machinery acts upon these genes, the structural variants and gene alleles are gonna affect which antibodies individuals develop. And so next we use these allele data sets as personalized allele databases to analyze the antibody repertoire sequencing data. A critical step in analyzing antibody rep-seq data is annotating each sequencing read with the correct V, D, and J allele. Here, as opposed to using a composite reference allele database, we use the personalized allele database generated from genomic data. And in contrast to previous human B-cell development studies using AIRR-C data, we sequence the antibody repertoire across development at an extremely high depth. Across individuals, we sequenced an average of 17,000 pro B-cell clones. Within the pre-B, immature, and naive B cells, we sequenced 123,000 to 265,000 B-cell clones for the heavy chain. For the kappa chain, we sequenced 9,000 to 35,000 B-cell clones. And for lambda, we sequenced 2,000 to 7,000 B-cell clones across the pre-B, immature, and naive B cells. And so the first question that we asked is, how does the heavy chain gene usage change across B-cell development? And so we answered this question by comparing the gene usage between cell stages. And what we observed is that the heavy chain gene usage is essentially staying the same across all B-cell developmental stages. The x-axis here is the gene usage at the pro-B-cell stage, and the y-axis is the gene usage at the pre-B-cell stage for individual E, and each point is a gene. And so across all individuals, the V gene usage correlation between the pro-B and the pre-B cells ranged from 0.92 to 0.98. And between the pre-B and immature B cell stages, it ranged from 0.95 to 0.99. And between the immature and naive B cell stages, it ranged from 0.85 to 0.93. Furthermore, across all the genes and individuals, the first quartile difference was at most negative 0.002, and the max third quartile difference was 0.005 between B cell stages, showing minimal changes to the heavy chain antibody repertoire. We also tested to see if any of these genes are consistently changing across all 6 individuals. Here, the y-axis are the IgH genes, and the x-axis are, sorry, the y-axis is individuals and the x-axis are the IgH genes. And each box is the gene usage difference between the pro-B and pre-B cell stage. And so we ran a paired t-test to determine which genes are consistently changing across all individuals. And so between the pro-B and pre-B stage, the usage of 6 IgH genes differ. Between the pre-B and immature, 6 IgH genes differ. And between the naive and immature B cell stage, the usage of 11 IgH genes differ. So in total, across all 3 comparisons, 19 or 20% of the IgH of the usage of IgH genes statistically differ. So given the high gene usage correlation found within individuals and the small magnitude of gene usage differences and the small number of genes that are changing between B cell stages, I believe that we could argue that the heavy chain antibody repertoire is now being altered extensively by the processes within B cell developments. This would therefore imply that the genome is playing a much more significant role in determining the heavy chain antibody repertoire than B cell development. And therefore that we should also expect to see a genetic effect on the heavy chain antibody repertoire at each B cell stage during B cell development. So to test this, we first started by performing a PCA on the IgHV, D, and J gene usage across all B cell types and individuals. On PC1, the points are separated by the B cell stages, and on PC2, points are separated by individuals. Next, we use the allele sharing distance, or ASD, which tries to capture how genetically different 2 individuals are. And here we're using IgH genetic variants to calculate ASD. And so by clustering ASD, or the allelic sharing distance, we observe that sample A is clustered away from everyone else, mirroring the patterns that are observed in the gene usage PCA space. Additionally, we observe that individuals CED are clustered together using ASD and in gene usage PCA space. To further show if individuals with more similar IgH loci also have a more similar antibody repertoire across B cell developments, we compared ASD to the repertoire dissimilarity index, which uses VD and J frequencies to measure how dissimilar the antibody repertoire is between 2 individuals. And we found that at all B cell stages, the ASD is nominally significantly correlated with RDI, providing more evidence that individuals with a more similar IgH loci have a more similar antibody repertoire throughout B cell development. Additionally, we sought to replicate the gene usage QTLs that we previously detected in the periphery. For example, we previously showed that individuals who are homozygous C have less IgH V366 in their naive antibody repertoire compared to individuals with the T allele at this particular SNP. And again, in fact, we found that the usage of 80% of IgHV genes are affected by a SNP. So out of the 28 GQTLs that we could replicate, 82% were replicated at a p-value less than 0.1 in at least 1 B cell stage. And 57% were replicated at a p-value less than 0.05. For example, individuals who are homozygous for a C allele at this SNP had higher usage of IgHV1-3 across all B cell stages compared to individuals with a G allele. Similarly, for IgHD 3-3, individuals with different genotypes at this SNP demonstrated different levels of IgHD 3-3 in their antibody repertoire at different stages. And many of these genes are in fact deleted in some individuals, which is what's likely driving the gene usage differences all the way from the pro-B cell stage. However, for the other genes that are not affected by structural variants, but whose usage are associated with IgH genetic variants, we previously showed that these genetic variants are enriched within regulatory regions implicated in regulating VDJ recombination. Here we identified a SNP associated with IGHV348 gene usage difference at the pro-B cell stage, and this SNP was also within transcription factor binding sites known to play a role in chromatin remodeling and VDJ recombination. And so what we're showing is that genetic variants are affecting the antibody repertoire all the way from the first stages of B cell development and are present throughout the development of the antibody repertoire. Of course, this means that if the heavy chain antibody repertoire is not changing, then the changes observed in the developing B cells must be occurring somewhere else. So next we looked at the development of the kappa and lambda antibody repertoire, which starts at the pre-B cell stage. And indeed, across individuals between the pre-B and immature B cells, the kappa V usage correlation ranged from 0.72 to 0.87. And the lambda V usage correlation ranged from 0.59 to 0.87, which is much lower than that observed in the heavy chain antibody repertoire, indicating much more extensive changes in the light chain antibody repertoire. Between the immature and naive B cells, the gene usage value showed much higher correlation, although across individuals, it was more varied in the lambda repertoire compared to the kappa repertoire. When we look at the gene usage differences between the pre-B and immature B cell stages, the first quartile is 0.01, and the third quartile is, the first quartile is negative 0.01, and the third quantile is 0.01, which is 5-fold higher than that observed in the heavy chain. Additionally, the gene usage of 52% of the kappa genes and 39% of the lambda genes significantly differed between cell stages, which is more than double the number of IgH genes that are changing within B cell development. Furthermore, when we compared the heavy kappa and lambda antibody repertoire between consecutive B cell stages using RDI or repertoire dissimilar index again, we observed that the kappa and lambda antibody repertoire are more dissimilar. And so what I believe we are observing is the significant effects of receptor editing, which is the only central tolerance mechanism that could alter the light chain antibody repertoire while keeping the heavy chain antibody repertoire. And so given the changes in the light chain antibody repertoire, we next wondered if we could see a genetic effect from the light chain loci on the light chain antibody repertoire. And so again, using ASD with genetic variants from the light chain loci, we did not find any significant association between the light chain loci and the light chain antibody repertoire. However, one possibility for this is that the light chain genetic effect is being masked by the heavy chain, meaning that the light chain loci do in fact dictate which V and J genes get selected for VJ recombination, but because the light chain pairs with the heavy chain and undergoes receptor editing, then the original VJ frequency gets obscured by chain pairing and or by receptor editing. And so in order to test this hypothesis, we tested for trans effects. Trans effects are effects from genetic variants that are present distally from the gene that is being affected. And so in this schematic, an IgH variant on chromosome 14 is associated with the usage of a kappa gene, IgHK V17 found in chromosome 2. And this effect could be mediated because this SNP is associated with this IgHD gene, with the usage of this IgHD gene. And so heavy chains with this gene might not pair well with this kappa gene, or create heavy and light chain chain pairs that are autoreactive and lead to receptor editing. And indeed, we found IgH variants associated with inter-individual light chain gene usage differences. For example, this SNP is associated with the usage of 4 IgHD genes and also associated with the usage of 2 kappa genes and the usage of 3 lambda genes. In total, we found that the usage of 36 and 38 kappa and lambda genes respectively were associated with IgH variants. And given that these variants are associated with multiple heavy kappa and lambda genes, we next tested to see if these variants could also be affecting the physiochemical properties of the antibody repertoire. And indeed, we identified variants associated with, for example, the charge of the naive heavy chain antibody repertoire and the acidity of the naive kappa antibody repertoire. And so, given that these physiochemical properties are dictated by the CDR3 amino acids, we next look to see if these variants were also associated with which amino acids are present within CDR3. Here I'm showing an example of a SNP that's associated with a fraction of different amino acids at different CDR3 positions. We were also able to replicate these associations in a larger cohort of 154 individuals. For example, individuals with homozygous for a C allele at this SNP on the right had a higher fraction of glycine at CDR3 position 3 than individuals with a T allele, and vice versa for the amino acid tryptophan at CDR3 position 9. And so to conclude, we've demonstrated minimal changes to the heavy chain antibody repertoire across B cell development. We've shown that genetic variants from the IgH locus are affecting the heavy kappa and lambda antibody repertoire from early B cell development. And these variants are affecting the antibody repertoire in the context of gene usage, physiochemical properties, and which amino acids are present within the CDR3. We also observed more changes in the kappa and lambda antibody repertoire across B cell development. We believe that, or we're showing signatures of genetic effects on the heavy chain being mediated most likely through chromatin changes that are affecting VDJ recombination, and that the genetic variants within the heavy chain loci are affecting the light chain antibody most likely through chain pairing and or the generation of autoreactive antibodies. And with that, I'd like to say thank you for listening and happy to take any questions.
Wow. I hope people have questions because I'm not smart enough to have a question. That was amazing stuff. Let's see, before we do questions, let me announce that Pam put in the information on the Porto meeting, June 3rd to 6th, and their posters can be accepted and travel awards. And then the next talk is going to be April 25th, Evelyn Georgia from Vanderbilt University and early career speaker at Marta Borowska from Stanford. And I'm going to scan here for questions. Don't have any immediate questions. And so these SNPs are all in the megabase, but in just all over non-code the Q, I guess QUTLs are, but not in coding regions or anything. They're mostly in upstream regions or all over the place. Or
yeah, that's right. The majority of the variants that we see associated with the differences in the antibody repertoire are within non-coding regions. That's why we think it's probably these variants are probably mediating all these effects through the regulation of VDJ, in addition to possibly other biological mechanisms.
Oh, Jamie may save me. Jamie?
I'm trying to. So that's really very interesting what you're finding, and especially where you've got this great data showing the effects of most likely of receptor editing. That's pretty wonderful. Yeah. So I guess what I wanted to ask you was about some of these mechanisms and how you might go about understanding them more thoroughly at the recombination and selection levels. And and also what that might have to do, with, especially, autoimmunity, if at all it has anything to do with it.
Yeah, so in terms of trying to tease out the regulatory mechanisms, one thing that is really lacking is essentially regulatory maps of the Ig loci. So usually in terms of like epigenetic regulation, you would run a whole bunch of different types of assays to understand which transcription factors are binding. You might run a Hi-C experiment to understand the 3D genome structure. But there has been, or at least we cannot find any data in humans showing what the actual epigenetic landscape looks like in IgH. And it's further complicated because, as Melissa showed, we showed that there's so much structural variance, there's so much the DNA being deleted. And so then the question also becomes, how does the epigenetic landscape look like in these loci across individuals if everyone has such a different IgH loci? So I think one of the critical things that has to happen is we have to start creating epigenetic maps at an individual level, which then could be also go beyond, well, that could also tie into trying to understand autoimmunity. And so if you imagine running an autoimmune study within the IgH locus and then finding a variant within the IgH locus that's associated with autoimmunity, the question is, okay, how is this variant causing autoimmunity? And one of the ways to answer that question is by looking at these essentially regulatory maps. This variant is affecting this change in chromatin structure, which is then going on to increasing the selection of this particular gene and putting that in the antibody repertoire. But if you don't have those regulatory maps, then it becomes really hard to map these variants and really find the causative reason as to why these variants are inducing autoimmunity or increasing the predisposition risk towards autoimmunity.
So just a note, Corey just put in the chat self-promotion bioRxiv paper that he and Eric and Oscar just published about why some of the short read epigenetic data isn't sufficient in this region. And I think it's interesting to consider. And Oscar, I mean, we're doing, for some of the individuals where we have really wonky Ig haplotypes, we are doing whole genome sequencing on the PacBio, and that at least will give us some 5MC signatures in the locus. So that'll be interesting to dovetail with what you're talking about.
Yeah, and I mean, they're starting to, I don't know if this is definitely going to happen, but they're starting to show for some autoimmune diseases that if they scan the antibodies, the sera, sometimes you can see these emerging autoantibodies well before symptoms develop. And as a result, you can start doing things to save the pancreas or whatever well before that. And so, it becomes, if you have, I don't know, what do you think? Do you think that people are ever going to be doing genomic sequencing and then focused deep sequencing on the IgH and loci and so forth? You think they're going to do that?
Definitely
for everybody? Because I mean, right now, like I'm in a thing that NIH is doing, and they take your blood, they store it, they do all this stuff with it, they give it to people, and they're also going to sequence my genome, but I don't think it's going to be sequencing to the depth that you guys do. So anyway, I mean, if they're doing that now, I'm just kind of wondering what that means in terms of being able to predict autoimmune predisposition and so forth. Yeah. And also, you're recombining your genes throughout your life, right? This isn't, and so is there anything that you can think of that would be changing that dynamic over time with aging, or have people found that?
Yeah, so for the first part, I definitely think that's the way the field is going. The NIH has this All of Us program where
that's what it is.
Yeah, they sequence 10,000 individuals with long read sequencing, And so there you actually might have the capability to look at the Ig loci. And so I think in the future, people are going to start putting the Ig loci into these polygenic risk scores, which will allow you to predict the risk of getting different autoimmune diseases.
Yeah, they're up to millions now with those.
Yeah,
it's crazy.
And there's more projects coming out where they're sequencing 100,000, or the plans are to sequence 100,000 individuals with long read sequencing. So I think that's the goal. I think the future is going to be looking at the whole genome to truly understand the predisposition risk to all of these diseases.
And what do you think about over the lifetime?
Yeah, I was thinking about. I've been thinking about that. I don't know because I can't think of a mechanism that would do that, because you have your common lymphoid progenitors. It would have some effect on the common lymphoid progenitors. I mean, you could think of a mechanism where you generate so much somatic mutations at these CLPs, which then might affect the way you're undergoing VDJ recombination and try to generate and then you ultimately generate a different antibody repertoire. That's one possibility. Yeah, I'm just not sure.
I was wondering.
Thank you.
Thanks. In a different kind of vein, but I mean, you're starting to get a large number of individuals, not at the pro-B, all these different stages, but in the locus, and you're working with Matt Pennell. I mean, can you start to look at like selective effects on these SNPs and selective sweeps and something like that, because it'd be interesting to start seeing if they're causing autoimmune diseases, what the selective effects are on these SNPs.
Yeah, no, for sure. I think that's definitely the goal. And people have shown it, not across the whole locus, but of variants like they could genotype within the IGH locus. They've shown that there's huge population differences. I think even the 1000 Genomes project that first came out showed like a SNP within the constant region that showed that people differed extensively across populations for this variant. But yeah, I think some results that William showed in his recent paper also showed that there was huge differences within the lambda loci across different populations. But yeah, I think that's the goal, or at least one of the goals.
Yeah, we spend a lot of time at that Dresden meeting running around in circles saying, is there any selection here? But it's gotta start answering that. I don't think I see any other questions, and we're a couple of minutes over, but those were, as I say, my brain is even fuller. Those are two great talks, and April 25th is the next seminar, and thank our two speakers again, and we'll see you in a month.